Dear students, in this class I would like to discuss semi-conservative model of DNA replication. So you know that replication is nothing but synthesis of daughter DNA from parental DNA is called replication, or duplication of DNA is called replication. So from the parental DNA molecule, daughter DNA molecule will be formed. That is called replication. So what is semi-conservative model of DNA replication? So whenever the DNA molecule undergoes replication, the two strands gets separated from each other and each strand can act as parental strand or template for the synthesis of daughter strand. Here, for example, this is the DNA strand. So for example, this is the DNA molecule. The DNA molecule consists of two strands. One strand runs from 5' to 3'. Another strand runs from 3' to 5' direction. So this is the DNA molecule. So if this DNA molecule undergoes replication, what happens here? Two strands are there. The two strands get separated first. First the two strands gets separated from each other and each strand can act as template here. Here this is the double-stranded DNA molecule. Watson and Crick proposed this double helical structure of DNA molecule, how the DNA looks like if it is double-stranded. So here the DNA molecule undergoes replication. When the DNA molecule undergoes replication, what happens? First the two DNA strands gets separated. Now this is the DNA molecule. The DNA molecule consists of two strands. This is the strand 1, strand 2. So when the DNA molecule undergoes replication, the two strands get separated from each other and each strand can act as template. This is first strand, this is the second strand. Both strands can act as template strand, or both strands can act as parental strand. For what? For the synthesis of new strand or for the synthesis of daughter strand. From this parental strand, one daughter strand will be formed. From this strand, another daughter strand will be formed. This is how the replication takes place. So, what is semi-conservative? Here, if the DNA molecule undergoes replication, what happens? First, the two strands get separated and a newly synthesized strand will be present in each DNA molecule. So, from one DNA molecule, two DNA molecules will be formed and each DNA molecule contains one old strand and one new strand. Here also, one old strand and one new strand. This is how the DNA replication takes place. It was termed as semi-conservative. In other terms, if we give the color, different colors, then you can understand very easily. So this is the DNA molecule. The DNA molecule undergoes replication. First the two strands get separated like this. One is 5' to 3', another one 3' to 5'. So these two are parental strands. From this parental strand, new strand will be formed. That is, from the parental strand, daughter strand will be formed in this direction. 5' to 3', 5' to 3'. DNA replication always takes place only in one direction. That we are going to discuss in detail in the DNA replication. So, the parental strand undergoes replication, or parental DNA molecule undergoes replication. Here the two strands get separated and each DNA molecule contains one old strand and one new strand, or one parental strand and one daughter strand. Here also same. So it was termed as semi-conservative model of DNA replication. The experimental proof for this semi-conservative model of replication is given by Meselson and Stahl. Now we are going to discuss that experiment. Experimental proof for semi-conservative model of DNA replication. Before that, you know what is semi-conservative, which means that each DNA molecule contains one daughter strand and one parental strand. What is conservative? The DNA replication is never and ever it is conservative. For your understanding, what is conservative? For example, this is the DNA molecule. If it undergoes, now I am discussing about conservative. In case of conservative, the DNA molecule undergoes replication. Here, the two strands gets separated and it can form the daughter strands from this one daughter strand, from this one daughter strand. Here what happens, these two daughter strands can combine together to form one DNA molecule, and the two old strand, this strand 1 and strand 4, this is 2, 3. These 2, 3 can combine together to form one DNA molecule, 1 and 4 can combine together to form another DNA molecule. This is called conservative. So this conservative model of DNA replication, it never and ever happens. We are going to discuss about semi-conservative. So what is the experimental proof for the semi-conservative model of DNA replication? So who has performed this experiment? Two scientists, namely Meselson and Stahl. Are the scientists who performed this semi-conservative model of DNA replication in a bacterium called E. coli. In E. coli. So E. coli is a bacterium, Escherichia coli. So they proved that DNA replication is always semi-conservative in bacterium called E. coli. Later it was shown in higher organisms like plants and human beings. First they have proved in the E. coli. These two scientists, Meselson and Stahl, have grown E. coli in a medium containing N15 nitrogen. Here they have used two isotopes, N15 and N14. So first of all, what is isotope? Isotopes are the atoms which are having the same atomic number but different mass number. So N15 and N14 having the same atomic number but different mass number. One is having N15 and N14. So you can call these are isotopes, isotopes of nitrogen. So in this experiment they mainly concentrated on the element or the atom nitrogen. You know that nitrogen is the basic element which is present in all the biomolecules. Nitrogen is present in DNA like under RNA or nucleic acids. Nitrogen is also present in proteins and other molecules. Here we are going to discuss about semi-conservative model of DNA replication. In that we are mainly concentrating on the DNA. So you know that DNA is a polymer of deoxyribonucleotide. So you know that in the previous class we discussed the structure of the nucleotide. Nucleotide is mainly composed of three components. You know that sugar, nitrogen base, phosphate. So nitrogen base contains nitrogen. So they mainly concentrated on nitrogen here. So in this experiment, in semi-conservative model of DNA replication, this Meselson and Stahl, they mainly concentrated on which element? Nitrogen. And in which organism they proved that DNA replication is semi-conservative? That is bacteria. What's the name of the bacteria? E. coli. In E. coli, they proved the DNA replication is semi-conservative. So here they used the isotopes N15 and N14 isotope. So you know that isotopes are atoms having the same atomic number, different mass number. So N15 and N14. Nitrogen is present in most of the biomolecules. Okay. First they have grown the bacteria in the medium which is having N15. And nitrogen, you know that for the growth of the bacteria, it requires medium. The bacteria requires medium. So you know that the medium usually consists of different nutrients. So without that nutrients, the bacteria cannot grow. So all the microelements and macroelements, different types of microelements and macroelements like carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, magnesium, calcium, different elements are there. All the elements are present in the nutrient media, but in this experiment, they provided all the elements along with nitrogen. Here, they mainly concentrated on nitrogen. So they have used the two different types of nitrogens, heavier nitrogen and lighter nitrogen. What is heavier nitrogen? N15 is the heavier nitrogen, N14 is the lighter nitrogen. You know that nitrogen is given in the form of NH4Cl, ammonium chloride. NH4Cl. Here nitrogen is there. So they have given the NH4Cl or ammonium chloride to the bacteria. For what? For nitrogen. So here they have used the two different types of isotopes, as I already said. They have used NH4Cl, that is heavier nitrogen, H4Cl, N14. So N15 media and N14. Shortly you can call it as N15 media and N14 media. So they used the two different types of media here. One media is having heavier nitrogen, that is N15. Another media is having N14. Now we are going to discuss in detail about the experiment. So how this N14 and N15 DNA can be separated. If the bacteria can be grown in N15 media, the DNA becomes N15. If the bacteria can grow in the N14 media, the DNA becomes N14. So how you can differentiate the N15 DNA from the N14 DNA? Usually there is a technique called cesium chloride density gradient centrifugation technique is used to distinguish the N15 DNA from N14 DNA. Here, what's the name of the technique? Cesium chloride, CaCl2, cesium chloride density gradient centrifugation technique. So, for what this technique can be used? To distinguish the N15 and N14. Here the name itself indicates density gradient. So this technique mainly depends upon the density. You know that N15 is more dense, having less density. N14 is less dense. So N15 DNA settles down at the bottom and N14 will be present on the top. For example, this is the centrifugation. So N15 DNA gets settled down here. N15. N14 will be present on the top. Why? So if you put both DNA, N15 DNA and N14 DNA, when you mix both N15 DNA and N14 DNA, and when you subject to cesium chloride density gradient centrifugation, in this technique what happens? All the N15 DNA settles down because they are having more weight or more dense. So it gets settled down. And N14 media is less dense. So this less dense particles will be present on the top. So this is N15 media, N15 DNA, this is N14 DNA. N15 DNA always gets settled down because they are having more weight or more dense. They are more dense. N14 media. N14 media is less dense when compared to the N15. So it is present on the top. So this is how you can easily distinguish the N15 DNA from N14 DNA by using cesium chloride density gradient centrifugation technique. So these are the basic things that you know before you know the what that they did the experiment. Now we are going to discuss the experiment. So what they did? First they have taken the bacterial cells. So you know that what is the bacteria they have taken here? E. coli, Escherichia coli. So they have taken the E. coli bacteria cells and they have grown in N15. Where they have grown? First they have taken the bacteria grown in N15 for many generations. That is usually 70 to 80 generations. So what is the generation time for the bacteria? 20 minutes. So they have grown the bacteria in N15 media for how many generations? 70 to 80 generations. So the DNA in the bacteria becomes N15 now. Now this is the N15 DNA. They have taken the normal bacteria. They have grown in N15 media. Just represent with blue only here. Bacteria, they have taken normal bacteria and grew in N15 media for how many generations? 70 to 80 generations. After 70 to 80 generations, the DNA becomes N15, N15. That is both strands. Here, why you represent N15, N15? This N15 represents first strand, this N15 represents second strand. Now this is the N15, N15 heavier DNA. Now they have taken this DNA as the first step in their experiment. So they have taken the N15, N15 DNA molecule and they grow the bacteria in the medium N14. They have taken the normal bacteria and they grow this bacteria in the medium N15, that is NH4Cl, N15 NH4Cl. Now, for how many generations? 70 to 80 generations. After 70 to 80 generations, what happens? The DNA becomes N15, N15. Now, they have taken this bacteria which is having N15, N15 DNA as the first step in their experiment. Now, they have transferred the bacterial cells which is having N15, N15 DNA into N14 media. Grew in N14 medium. So what happens? You know that whenever the DNA molecule undergoes replication, what happens? First the two strands get separated. These are the two strands gets separated like this. The two strands gets separated. Now each strand can act as parental strand or template strand. From each strand, daughter strand will be formed. Now these two scientists, Meselson and Stahl, they have grown the bacteria in N14 media. So the daughter strand will be N14. Here blue color represents the daughter strand. Why it is N14? Because they have grown in the media N14. That's why the DNA becomes N14 here. That nitrogen is incorporated in the N14 is incorporated in the daughter DNA strand. Okay. Now this is the hybrid DNA. N15, N14 hybrid. N15, N14. You understand? First they have taken the normal bacteria, they have grown in the media which is having N15 for 70 to 80 generations, so that all the DNA that is present in the bacteria becomes N15, N15. They have taken this DNA as the first step in their experiment. Now this is the bacteria which is having the DNA. Now they have grown the bacteria in the media which is having N14, so that after 20 minutes what happens? What is the generation time for the bacteria? 20 minutes. So after 20 minutes what happens? One bacterial cells becomes 2. So obviously the DNA undergoes replication. One DNA molecule becomes 2 DNA molecules. So here the two strands gets separated, the two parental strands gets separated, and each strand can act as template or parental strand for the synthesis of daughter strand. So here the red color represents the parental strand. The two parental strands gets separated. Here these two strands, and the daughter strand will be formed. Here N14, daughter strand is N14 here. Why? Because you are growing the bacteria which is having N15 DNA is growing in the N14 media. The two scientists, they have grown in the N14 media. That's why the daughter strand becomes N14 here. So now that is the hybrid DNA. What hybrid? N15, N14 hybrid. Now what they did? They have transferred the bacterial cells after 40 minutes. This is after 20 minutes. So after 40 minutes what they did? They have transferred into the media which is having again N14. So in the first generation also, this is the first generation. Now this is the second generation. Now they have grown this bacteria in the media which is having again N14. Before the first generation what they did? They have grown in N14. Again they got the N15, N14 hybrid DNA molecules. Again this N15, N14 hybrid DNA molecules, the bacteria which is having these hybrid DNA molecules were grown in again N14 media. So what happens? Here the two strands gets, this DNA molecule can act as parental strand. The two strands can act as parental strand. Here also the two strands can act as parental strands. So first the two strands get separated. This red and blue color strands, they get separated. The first DNA molecule, it gets separated. Now coming to the second DNA molecule, the two strands gets separated. Clear? So each strand can act as template strand or parental strand. Now where they have grown these hybrids, in which media? N14 media. So the daughter strand will be N14. All the daughter strands will be N14. N14. So this is N15, N14, this is N14, N14, N15, N14, N14, N14. Clear? This is the second generation. So what happens in the second generation? 50% of the DNA molecules are hybrid. 50% of the DNA molecules are lighter DNA. So here 50%, that is this and this DNA molecule are hybrid. 50% of the DNA molecule are hybrid and the 50% of the DNA molecule are lighter DNA molecule in the second generation. What happens in the first generation? 100% are hybrid, but in the second generation, in the first generation 100% are hybrid. In the second generation 50% hybrid and 50% are lighter DNA molecules, which clearly proves that the DNA replication is semi-conservative. If the DNA replication were conservative, what would be the result? In the first generation, definitely heavier DNA molecule will be formed, but it is not happened. Only hybrid is formed. If the replication was conservative, definitely what would be the result? So this N15, N15 DNA molecule will be formed here and N14, N14 molecules would be formed, but it is not happened. In the second generation also N15, N15, these two red color strands can combine together to form one DNA molecule if it would be conservative. So here in the first generation and in the second generation, you didn't get the heavier DNA molecule, both strands in the one DNA, both heavier strands in the one DNA molecule. So which clearly proves that DNA replication is semi-conservative. And is it possible to see the DNA with the naked eye? No. Then how they concluded this hybrid is formed in the first generation and both hybrid and lighter DNA are formed in the second generation? How they got? So they performed, I already said, they performed the technique called cesium chloride density gradient centrifugation technique. In this experiment, they used the centrifuge tubes, centrifugation process they have used here. So this is the centrifuge tubes in the first generation, this is in the second generation, this is in the third generation. Now, so N15, N15 DNA molecule, the first initially, so band is formed at the bottom, red color band. Why the band is formed at the bottom? Why not at the middle and top? Because N15, N15 is the higher DNA molecule. The two strands are N15, N15. In the DNA molecule, the two strands are N15, N15. So N15 is the heavier nitrogen. So the DNA molecule which is having N15, usually it gets settled down at the bottom. So band is formed at the bottom like this. In the first generation, what happens? The band neither forms at the bottom nor forms at the top. Why? No heavier DNA, no lighter DNA is formed in the first generation. 100% hybrid. This is hybrid DNA molecule. This is hybrid DNA molecule. So band is formed at the middle. So band is formed at the middle. In the second generation, what happens? 50% of the DNA molecule is hybrid and the 50% of the DNA molecule are lighter. So the lighter DNA, for the lighter DNA molecule, the band is represented at the top here. For the hybrid DNA molecule, the band is represented at the middle. So based on that, they concluded that the DNA replication is semi-conservative. So after Meselson and Stahl, Taylor and colleagues, another scientist, they performed an experiment in Vicia faba. It's a one type of beans plant. In that, they performed the experiment whether the DNA replication is semi-conservative or not. So in plants also, they got the same result, but they did not, these scientists, Taylor and colleagues, they did not concentrate on nitrogen, they concentrated on radioactive thymidine. They concentrated on radioactive thymidine. They performed the same experiment, but they did not concentrate on nitrogen, they concentrated on radioactive thymidine. You know that thymine is present in DNA but not in RNA. So they concentrated on thymine. So what they got the result? The same result they got. That is, the DNA replication is semi-conservative. So heavier DNA can be represented with red color. The two strands can be represented with red color. So it is heavier DNA. Hybrid DNA can be represented with one strand with red color, another strand with blue color, and the lighter DNA can be represented with blue color only. The two strands will be blue color. So heavier DNA, this is heavier DNA, this is hybrid DNA, this is lighter DNA. One thing you should remember here, N15 and N14 are not the radioactive isotopes. They are just isotopes. They are not the radioactive isotopes. Here thymidine is the radioactive isotope I said. Here N15 and N14, N15N15 or N14N14 or N15N14. They are not the radioactive isotopes. Here the N15N14 are the isotopes. So what are isotopes? Isotopes are the atoms which are having the same atomic number but different mass number. They are not radioactive. So N15 and N14 are not the radioactive. Important. So with this, semi-conservative model of DNA replication is completed.